all right, it will affect your data. So you will have an inaccurate data, okay, if the cell enter the flow cytometry in a clump condition. Okay, so it is very important for you to ensure cells that enter the flow cyto machine is in a single cell. Okay, so the measurement for the flow cyto involves cell size. So meaning that this flow cyto can measure the cell size, it can measure the cell granularity, amount of cell components, okay, such as total DNA, newly synthesized DNA, and gene expression, amount of specific surface receptors, okay, amount of specific intracellular proteins, okay, and transient signaling events in living cells. Okay, so some of these measurements, okay, it needed for the cell to be stained. Okay, cell tu kena diwarnakan dulu. The cell has to be dyed or stained. Okay, dia kena dye, dia kena diwarnakan, stained. Okay, okay, once it is stained, okay, then only you can measure the intensity of the cell. Okay, depending on the amount, depending on the protein of interest. Okay, okay. So there are two major principles, okay, measurement of physical properties and measurement of antigenic properties. Okay, so meaning that you can measure the physical, the size, the granularity of the cells, okay, and you can also measure the antigenic properties. So usually these antigenic properties is what is the method that it involve staining method. Okay, they involve dye method. Maknanya you kena warnakan the cell. Okay, alright. So unique ability. So this full cycle, it can give you rapid analysis, okay, of thousand cells. So usually, okay, when we use the full cycle, when we want to acquire 10,000 cells, okay, it involves several thousand cells, okay, 10,000 to 20,000 to 30,000 thousands, okay, it depends on the cells. Usually for adherent cell, you tahu kan adherent cell? Adherent cell tu adalah sel yang dia attach, okay, at the bottom of the flask, okay, so itu kita panggil adherent cell. Okay, usually adherent cells, okay, the size of adherent cells is bigger. Okay, dia juga ada cell of what we call floating cells or non-adherent cell. So these non-adherent cells, okay, the cell size is very small. Okay, so usually for the cell size that is very small, you have to acquire more. Okay, you have to acquire more. For example, macam PBNC. PBNC is alamak, apa PBNC ya? It's a blood, okay. It can be obtained or it can be isolated from the blood. Okay, so the size of the cells, okay, they are small, okay, compared to the adherent cell. Okay, adherent cell dia lebih besar. Okay, and for adherent cells, dia ada possibility to clump, okay, more than the non-adherent cell. Okay, so this is the unique ability of the Flowcyto. Okay, it can analyze thousands of cells. Okay, simultaneously illustrate multiple antigens. Okay, now, or as I mentioned earlier, when you talk about multiple antigens, okay, measurement of antigen properties. Okay, so this one, macam I kata tadi, they involve staining cells. So the cells have to be stained, okay, before you can run through flow cytometry. Okay, so it can also measure different property of cells, okay, able to categorize and quantify. Okay, now this categorized cells ni, okay, for example, you have the PBMC. So in the PBMC, there are so many populations. Okay, maknanya dalam PBMC tu, okay, there are many types. Okay, so what can this Flowcyto do? Okay, this Flowcyto can segregate, okay, segregate the population of the cells. Okay, dia boleh segregate dan dia boleh quantify. Alright, so even able to separate out subpopulations. So this is what it means by that. Okay, PBMC is peripheral blood mononuclear cells. Okay, so in the peripheral blood mononuclear cell, that's in PBMC. Okay, dia ada beberapa subpopulation. Okay, so usually you cannot separate it by your own. Okay, because of what do you call it, it is too small, and obviously you cannot see, okay, even through the microscope. So what you can do is, okay, you extract out, okay, the PBMC, okay, and then you run through Flowcyto, okay, and then what does this Flowcyto do? Okay, this Flowcyto is going to subpopulate, okay, the cells, extract the cell, isolate the cells, okay, and from there the Flowcyto itself is going quantify, okay, the cell based on the subpopulation, okay. So itu kelebihan flow cyto, okay, that's the advantage of flow cytometry. All right, so it can also tell you, okay, what cells are in there, okay, and yes, okay, flow cyto can be used to quantify the cells, okay. All right, so how could you separate your samples of white blood cells, okay, into subpopulations, okay, and what characteristic of the cell could you use, and how about if they had no color? Okay, so these answer, okay, these questions, okay, we are going to answer, okay, as we go along. All right, so this is the standard differentials. All right, so this one, if let's say you use the conventional method, okay, the conventional method is where you did not use, okay, the flow cyto. Okay, what you can do is draw a sample from a patient, okay, and then you smear blood on slide, okay, and then you stain with differential right stain, okay, you still have to stain the cell, okay, and then you have to count. Okay, so this method is very it looks simple, okay, it looks simple, but it is tedious, okay, because you really have to strain your eyes, okay, and see the size of the size of the cells, okay, the shape of the cells, okay, and you must be able to segregate, okay, the cells on your own, okay. So it is very, the process is very tedious, okay, and you have to make sure the amount of cells must be, not more than roughly hundreds, okay, because if it's more than hundreds, then you tend to make mistake, okay, because of course you punya mata tak boleh bekerja dalam tempoh masa yang lama, especially when you really focus, okay, on the type of the cells or the shape of the cell. So it's going to be very difficult. All right, so why do we need this technology? All right, look here. All right, so introducing the particle or cell into the flow cell. Okay, so flow cytometry, all right, is performed on particles in suspension, okay, such as cells or nuclei. Okay, so the inner column of the flow cell consists of liquid sample. Okay, the outer column of sheath fluid, okay, controls the diameter of the sample column so that it narrows and isolates single particles. Okay, the single particle passed through a laser beam. The light scatter is detected by a photodetector. Okay, so this will be a bit difficult, okay, because you didn't see the machine itself, okay. So okay, so this is where the sample will come through, okay, the flow cyto. All right, so again, okay, the samples contain cells, okay, and the cells must be in single cell. Okay, so whenever the cell enter the flow cyto, always remember that it must be in a single cell. All right, so this is the sheath fluid. Okay, so the sheath fluid will be at this area. Okay, it will be scattered at this area. All right, so what happened is that, okay, this is where your sample will be sucked into, okay, sucked into, okay, and then it will pass through a laser beam. Okay, so this is the laser beam. Okay, so what happened is that this laser beam will shoot at the cells, okay, and whenever it pass through the cells, okay, it will form a forward and side scatter. Okay, so fluorescence emitted from stained cells detected. Okay, forward and side scattered light from all cells will be detected. Okay, so basically this is the general principle of the flow cyto. Okay, so you have the cell. Usually the cells will be in a flow cyto tube. Okay, flow cyto tube tu dia lebih kurang macam ni. Apa ya? Okay, lebih kurang macam ni lah, lebih kurang macam ni. Okay, flow cyto, tapi ni gemuk kan. Okay, dia lagi kurus lagi. Okay, so what happen is that you'll have kejap macam ni lah, macam ni. Okay, so example ini lebih kurang lah, ini adalah Flowcyto cells. Katakanlah this is the Flowcyto tube, okay, tube of the Flowcyto. Okay, so what happened is that, okay, this tube will be introduced in the Flowcyto machine. So usually the Flowcyto machine dia macam ni. Okay, dia macam ni. Alright, dia akan ada dia akan ada macam satu jarum, okay, very small one, okay, where you will introduce, okay, this tube, okay, into the Flowcyto punya needle, okay. So what happened is that, okay, this Flowcyto needle, okay, will suck the cells, okay, inside the Flowcyto. Okay, so the cell will be sucked into the cells will be sucked into the Flowcyto. Okay, nanti dia akan ada di sini. Okay, it will pass through here and that it will pass through the laser beam. Okay, so if you look here, nampak tak? Cell pass through in a single form. Okay, dia tak boleh in a clump form. Okay, kalau dia clump form nanti, okay, kalau dia clump form, contohnya macam ni, contoh macam mana clump form dia? Contoh sekejap. Nah, susah lah, kesian kan you all. Okay, contoh katakanlah this is the clump cell kan. Okay, so of course inside here also got cell. Okay, so outside outer layer dia pun ada cell. Okay, so what happen is that if these clump cells, okay, pass through the laser beam, okay, the laser beam, the intensity of the stain cells, okay, dia tak boleh nak baca di bahagian dalam, okay, because it's already clump. Okay, so it can only be read, okay, at the through the outer layer. Okay, so the result that you've got, okay, won't be accurate. Okay, faham tak? Faham ke tidak? Saya susah hati takut you all tak faham sebab you all tak pernah tengok lagi Flowcyto machine ni. Faham, okay. Alright, tak faham tanya ya? Okay. Alright, so this is what happened. Okay, when the laser, okay, contohnya, okay, contohlah ini cell. Okay, so ini laser. Okay, so ini cell. Okay, so ini laser. So what happened is that, okay, when the cell pass through the laser, okay, laser tu dia akan ada cahaya kan? Okay, akan ada cahaya. Okay, so bila cahaya tu lepas keluar daripada cell, okay, it will form a forward scatter. Okay, so this is the forward scatter and this is the side scatter. Okay, the forward scatter and the side scatter. Okay, or it can be like this. Okay, the forward and the side. Okay, all right. Okay, type of light scatter. Okay, so as I mentioned earlier, again, okay, so this is the laser. Okay, and this is the cell. Okay, so when the cell pass through the laser, okay, so there will be light. Okay, light emitted to the cell. Okay, and then pass through the cell. So when pass through the cell, dia akan bentuk macam ni. Okay, so this is the forward, this is the side. Okay, so side light scatter, okay, they're scattered at a 90-degree angle, okay, from particles define internal complexity. Okay, so what does the side scatter define? Okay, dia akan it will define the internal complexity and granularity of the particles. Okay. Okay, so examples, okay, neutrophils and eosinophils produce a great deal of side scatter due to their cytoplasmic granules. Okay, so usually this side scatter, okay, dia akan measure the granularity of the cells. Okay, granularity tu maknanya the shape, okay, of the surface of the cells. Okay, so that's the granularity. Oh, sorry. Okay, so forward scatter that continue in the forward direction relates to the particle size. Okay, so it will measure the size of the cells. Okay, so large cells, okay, such as monocytes, okay, and neutrophils produce more forward scatter compared to red blood cells, okay, and normal limbs, okay, because these kind of cells, okay, normal RBC and normal limbs, dia punya size dia lebih small. Okay, compared to the monocyte and the neutrophils, alright. Therefore, okay, the presence of the side scatter, okay, and forward scatter will produce a more accurate data, okay, compared to the conventional method where you have to withdraw the samples from the patients and then you have to stain and then you kena smear on the on the slide, lepas tu you kena tengok pula bawah microscope, so it's very tedious. Okay, the process is very stressful, okay, compared to just you just have the cell sucked into the Flowcyto will do everything for you. Okay, but the most important thing is you should know what type of stain you should use. Okay, all right. So light scatter plot. Okay, so this is the forward scatter and this is the side scatter. Okay, so all these populations, okay, it is actually obtained from PBMC. Okay, I mentioned earlier PBMC. PBMC is peripheral blood mononuclear cells. So in the PBMC itself, okay, there are so many subpopulations. Okay, so these are the examples of the subpopulations in the PBMC. Okay, so if you run through Flowcyto, okay, so it will subpopulate for you. All right, so it will segregate the cells and quantify the cell for you. Okay, so it will form lymphocytes, basophils, eosinophils, neutrophils, monocytes. So how this done? Okay, this is done based on the data obtained from the side scatter and the forward scatter. So always remember the side scatter, it defines the granularity, okay, of the cell. Meanwhile, for the forward scatter, it defines the size of the particle. Alright, so jelas ya? Okay, I'm going to go very slow, okay, because you tak tengok mesin ni, so I sangat risau. Okay, so let's look at the fluorochromes. Okay, so fluorochromes are fluorescent compounds, okay, that can be bound by antibodies, okay, to the particles in the sample. Okay, so example of the fluorochrome, you have the FITC, you have the PE, propydium iodine, acrylic orange. Okay, so the fluorochrome is the one I told just now when I talk about staining cell. Ingat tak? I cakap pasal staining cell. I kata before you analyze the cells using Flowcyto, you have to stain the cells. Okay, so the stain the cells ni lah dia fluorochromes ni. Okay, so fluorochromes are used in immunophenotyping, okay, cell differentiation and enumeration, reticulocyte counting and DNA analysis. Okay, so that is, so it's very important. Okay, when you want to purchase any stain cells, okay, any what do you call it, uh, any fluorochromes for you to stain the cells, okay, you must know what do you want to look at, okay, whether you want to look whether you want to observe the immunophenotyping, or you want to look at the cell differentiation, or if you want to do the DNA analysis. So there are more actually, okay, so you have to be very careful when you select, okay, the staining. All right, okay, so detection of fluorochromes. Okay, so first, the laser beam excites, okay, fluorochrome attached to particles at one wavelength. Okay, so fluorochromes emits light, okay, at different wavelength. Okay, and then what happened is that, okay, ni cerita dia macam ni lah yang tadi saya ceritakan tadi tu. Okay, so you have the laser beam, okay, and you have the cell. Okay, so cell pass through the laser beam. Okay, so laser beam will excite the fluorochromes. Okay, so what happened is that, okay, the fluorochromes emits light at different wavelengths. So what happened is that you've already stained the cells. Okay, you've already stained the cell with whatever stain you want to stain. Okay, so some of the wavelength will be absorbed. Okay, and some of them will pass through. Okay, so whatever is passed through, okay, is directed to the photomultiplier tube, okay, that detects and quantify the light. Ini kan konsep dia lebih kurang macam spectrophotometer. Pernah dengar kan spectrophotometer during your first year? Konsep dia lebih kurang macam tu. Okay, tapi this one is the advanced one. Ini adalah high end machine and it's very expensive. Okay, so once the light, okay, reach the photomultiplier tube, okay, it will be how do you call it, converted, okay, by the CPU into computer software, okay, and analyze the data and generate this scatter plot. All right, but as I told you, the first thing that you need to do is you need to find, or you need to stain the cells, okay, with the correct staining. Okay. Okay, so immunophenotyping uses antibodies and antigens. All right, so immunophenotyping is the identifications, okay, of antigens, okay, using detection antibodies. Okay, monoclonal antibodies are manufactured using myeloma cells, okay, to bind with a single portion of an antigen. Polyclonal antibodies are made by injecting antigen into animals and are directed against multiple portion of an antigen. Okay, and most immunophenotyping studies detect cell surface antigen. Okay, so these are actually, okay, the antibodies and the antigens, that you use, okay, when you want to be immunophenotyping. Okay, so that's why, okay, before you start any project, okay, it's very important for you to read through. Okay, you have to read through journals, okay, related to your project, okay, for you to know what types of antibody that you can use. Okay, either it is monoclonal antibody or polyclonal antibody. Okay, so this is very basic. Okay, whatever I taught you, whatever I told you today, okay, it is very basic. Okay, however, whenever you want to start doing your project, okay, it is very important you identify, okay, what types of antibody that you want to use, and what types of data, or what types of protein of interest yang you nak. Okay, by then only you know what type of antibody can be used. All right, so commercial antibodies are grouped in clusters of differentiation. Okay, so this is what we call CD markers. So it acts as a marker. Okay, for example, katakanlah, okay, for example, you want to look at the presence of K space, example, so you want to look at the presence of K space. So how can you be so sure, okay, that the K space exists, okay, in that particular cell? Okay, so what you can do is, okay, you can have a control. Okay, so usually the control is the one is the untreated. Okay, or for this antibody, okay, you can have the control, okay, where what you call it as a CD markers. Okay, so meaning that, like it or not, okay, that particular case spaces, okay, will you can, it can be seen, okay, through your data, because it is a marker. Okay, so CD designations or CD marker, where a group of antibodies are recognized by the same antigen. Okay, so nowadays, okay, it is commercially available. Okay, meaning that if let's say you want to look for a specific proteins, okay, so you're not sure whether the the stain that you use, okay, or the protein that you use, or the antibody that you use are going to work. Okay, so what you can do is you can purchase the marker. So CD marker antibodies labeled with fluorochromes, okay, have been developed to bind with antigens of identifiable cells. So CD designations are cell markers, okay, for example, CD4, or where it is a helper to T cells, CD8, okay, which is cytotoxic to T cells. All right, so immunophenotyping with Flowcyto. Okay, so this is what happened, okay, in a CD marker. Okay, the manufactured antibody, okay, usually manufactured antibody ni memang kita customize dia. Okay, kita make sure the antibody itself can have the particular signal that you want. Okay, so that's what we call it manufactured antibody or CD marker. So the manufactured antibody, okay, is attached to a fluorochrome. Okay, this one, attached to a fluorochrome, okay, such as FITC, and then added to the sample. All right. So if the cells has the antigen that the antibody specifically binds the, if the cells has the antigen that the antibody then the antibody specifically binds, okay, the antibody and the fluorochrome attached to the cells. Okay, so let's say this is the cells, okay, and at the surface of the cells, okay, there is the antigen. So this is what we call antigen of interest. Okay, and then you have the antibody that attached with the fluorochromes. Okay, so what happened is that if this antibody is complementary to the antigen, okay, the site, the binding site is complementary to each other, okay, then what happened? It will bind to it, okay, together with the fluorochrome. So that is why, okay, when the cell, okay, which has been seen, okay, pass through the laser beam, okay, this laser beam will emit its signal, okay, and reach the fluorochrome. Okay, that is when you can have the readings. Okay, so when the cell or antibody or fluorochrome complex passes through the laser beam, the fluorochrome is excised, okay, and fluoresces at the measurable wavelength. Okay, and then what happened is that they can pass through, okay, in a side scatter and forward scatter form, okay, and then it will reach the photomultiplier. Okay, so the photomultiplier will reach the CPU. Okay, so in the CPU itself, you've already installed the software. Okay, and then the software will plot, okay, will do the scatter plot for you. Alright, so the computer software connected with the flow cytometer generate a histogram, okay, that visually represent the cells that is present. Okay, so no target molecule, meaning that there is no fluorescence. Okay, there's no fluorescence. Okay, so therefore it is very important for you to understand the concept of a lock and key. Kalau macam dalam enzim kan kita ada lock and key kan. Okay, the complementary is very important. Okay, the complementary at the binding site is very important. Okay, so the antigen, okay, must be complement with the antibody. So when it is complement, okay, then only it can bind to each other. So upon binding, okay, when the antibody, okay, that has the fluorochrome, okay, bind to the antibody, the fluorochrome will bind to it too. Okay, so that's why when it pass through the laser beam, okay, the signals emitted from the laser, okay, will excite, okay, whatever fluorochromes that you've stained. Okay, and then when it pass through, it will pass through in a side scatter and forward scatter. Okay, so whatever that pass through will reach the photomultiplier. Okay, and photomultiplier will convert it into a scatter plot. Okay, alright. Faham tak so far?
Faham, doktor.
Good, alright, thank you. Okay, now the many parts of flow. Okay, so as I told you before, Flowcyto is a very big machine. Okay, it is a high-end machine and it is a very expensive machine. Okay, so obviously it has many parts. Okay, so first of all is what we call flow basic. Okay, where it involves the experimental design, sample preparation, choosing proper instrument, and setting up the instrument. Okay, now this is what we call flow basic. Okay, flow basic is actually the preparation before you do the experiment. Okay, so you must know what is your experimental design. Okay, and then how should you prepare your sample? Okay, what type of stain should you use? Okay, what type of antibodies should you use? What type of antigen should you use? Okay, and then collecting proper data and interpreting the data. Okay, so when you have already setting up, okay, the instrument, okay, what happened is that you'll have the scatter plot. Okay, and then you have to collect the data. That's what we call data analysis. Okay, you have to collect the data and then you have to interpret the data. Okay, and then after that, okay, graphic presentations and publication as well as sorting out. So that's what we call specific application courses. Okay, so uses of flow cytometry. So this flow cytometry has been using for centuries. Okay, it can be used, okay, for so many things, okay, which you can read here later. Okay, and usually projects, the project or research project with Flowcyto, okay, the possibility for it to be accepted, okay, in a publication is very high. Okay, because the accuracy, okay, is also very high. Okay, they're very accurate. Whatever data that you got from the Flowcyto is very accurate. Okay, and you also have to remember, whenever you run the Flowcyto, you tak run satu kali. Okay, you have to also run several times. Okay, and several times maknanya tak semestinya 3 kali. Okay, it depends on the it depends on whether your data is fluctuate or not. Okay, so if your data, let's say you buat experiments pertama, eksperimen kedua, okay, you do first experiment, second experiment, third experiment. Okay, all the first until third experiment, the data will very fluctuate. Okay, fluctuate, you got fluctuating data. Okay, can you accept it? Kalau you tak boleh nak accept it, then you have to repeat. Okay, so meaning that let's say you buat first, second, third. Okay, kan kita katakan, okay, all data you kena buat in triplicate. Tapi kalau data you triplicate, untuk tiga triplicate tu, it's very fluctuative. Obviously you cannot use it. Okay, because it will produce a very high error bar. So very high error bar shows that your data is very inconsistent. Okay, so those inconsistent data, okay, kita tak terima. Okay, we won't accept it. Okay, so that is why you have to be very careful in selecting or in designing your experiment. Okay, and this flow cytometer, dia punya penggunaan dia very expensive. Okay, the antibody is expensive, the antigen is expensive, and the flow cytometer and the machine itself, okay, certain lab they charge it. Okay, satu sampel dia charge dalam berapa 10 ke 20 ringgit. Okay, so usually for you to use a Flowcyto, dia tak semestinya satu sampel tau. Sometimes it can be sampel yang berpuluh-puluh. Okay, you will have many samples. Okay, so you imagine, let's say you have 10 samples. Okay, so 10 samples, 10 samples times with 20, itu saja dah 200. Okay, itu baru satu replicate. Okay, you tak kira lagi 3 replicate. Okay, kalau 3 replicate ni, tiga-tiga very fluctuative, okay, you have to repeat it. Itu je so it is very costly. Okay, so that's why whenever you have the data with Flowcyto, okay, usually it is easily acceptable. Okay, dalam mana-mana paper, mana-mana journal, selalu senang nak acceptable. Okay, alright, accepted. Okay, okay, so let's look at the mechanic of a flow cytometer. Okay, so for the flow cytometer, okay, tadi kita cakap now, hold on let me go. Okay, now this one is about the preparation. It involves the preparation, okay, while doing it and after doing it. Okay, this one, okay, this one is about the flow cytometer itself. Okay, the mechanic part of the flow cytometer. Okay, so it consists of three, okay, where you have the fluidics, okay, optics, and electronics. Okay, so the fluidic part, okay, is where the cells in suspension are brought in single file passed. Okay, so in a way, okay, when you talk about fluidics, okay, it is very important for you to make sure to ensure the cell that enter the flow cycle is in a single cell. Okay, ini dia punya general term lah. Okay, for optic, a focus laser where they scatter light and emit fluorescence that is filtered and collected. Okay, for the electronic part, okay, it will be converted to digitized values, okay, that are stored in a file for analysis. Okay, so in a very simple way, when you talk about Flowcyto, there are three mechanics part. Okay, so first of all is the one that I told you when you have the Flowcyto tube, okay, and then you introduce in the Flowcyto machine. Okay, so what happened is that whatever cells in there will be sucked into the Flowcyto. Okay, so that is the fluidic part. Okay, and then the optic part, okay, is where you have the laser. Okay, you have the laser and you have the cells. Okay, so the cells with the fluorochromes, okay, will pass through the laser. Okay, so that is the optic part. And then the last part is where you have you've already had the scatter light, okay, where the scatter light reach the photomultiplier and then it is converted into the analysis part. Okay, that is the scatter plot. Okay, so that is the electronic part. Okay, so it's very easy. So there are three important parts. Okay, okay, so okay, kejap. Okay. Okay, sorry. Alright. So this is the simplified version of flow cytometer. Okay, so this is the one, okay, that I told you about the ni. Okay, the flow cyto tube. Okay, ni okay, ini flow cyto tube. Ni okay, flow cyto tube. Okay, and then introduced to flow cyto. Okay, so this is the needle. The needle. Okay, masukkan dalam ni. Alright, and then what happened is that the cell will pass through here, and reach here. Okay, so this is the fluidics part. Okay, this is the fluidic part. Okay, kemudian dia datang sini. Okay, so this is the laser. Okay, this is the laser. So when it enter this part, okay, this is what we call optic part. Optic kan? Okay, optic part. Okay, can you see the laser shoot at the cells. Okay, and then the light will be scattered. Okay, the light will be scattered according to the fluorochromes that you use to stain the cells. Okay, and then what happened is that this scattered light, okay, will reach the photomultiplier. Okay, so the photomultiplier will convert it. Now the latter part is actually the electronic parts. Okay, so senang kan? Okay, itu sangat-sangat senang. So this is the simplified version of a flow cyto. Faham kan? Faham ke tidak? Tak apa. Kalau you tengah makan tu you type je.
Faham, doktor.
Comel. Okay. Alright. Okay, so let's look. Okay, so this is the fluidics part. Okay. Okay, thank you. Thank you, Lisa. Okay, so you have the sample. Okay, and then in the sample, you have the sample tube. Okay, so what happened? This is the needle. Okay, that I showed you just now. Okay, suck the cell. Okay, and then the cell will flow to this area. Okay, so this is the area of where you should have the laser. Okay, laser. Okay, kat sini lah laser dia. Okay, so the laser will pass through single cells. Okay, and then now bila laser tu dah pass through single cells, where should the single cells go? Okay, macam ni kan. Okay, so kita ada laser. Okay, dan kita ada cell. Okay, so when the cell dah pass through the laser, okay, you've already had the readings. Sekarang ni cell ni nak pergi mana ni? Cell ni nak pergi mana? This cell will go to waste tank. Waste tank. Maknanya dia akan jadi waste lah. Okay, so that is why, okay, this Flowcyto, it needs maintenance. Okay, dia perlukan maintenance. Okay, why? Because if let's say it's heavily used, okay, the waste tank will be full. Okay, so kita kena empty dia selalu. Alright? So what happened is that there will be vacuum. Okay, but you have to understand that this Flowcyto, it needed maintenance. So maintenance is very important. So what does this chest tank do? Chest tank, okay, will control the pressure. So pressure dia akan control dari sini. Okay, pressure akan control dari sini. So chest tank dia akan control pressure at this area. Dia akan control pressure at this area to make sure the window, ni window kat sini dia punya bukaan. Dia punya macam mana cakap sel tu kan macam mana saya nak buat dia? Saya kalau boleh nak you faham betul-betul. Okey, contoh. Okey, contohnya lah, contoh the cell pass through. Okay, contohnya the cell pass through here. Okay, the cell pass through here. Okay, before it can reach the laser beam kan. Okay, so what does this chest tank do? Okay, this is chest tank. Okay, dia akan control this area. Okay, dia akan sama ada narrow it, okay, or make it bigger. Okay, so that is what the chest tank do. Okay, at this area, chest tank at this area. Okay, dia akan control the window. Yang ini yang kita panggil window ni. Okay, dia akan control the window. Okay, sama ada dia akan widen the window or dia akan narrow the window. Okay, alright. Okay, so fluidic sample differential. Okay, so this is the one. Different in pressure, okay, between sample and chest. Okay, so this will control the sample volume flow rate. Okay, so the greater the differential, the wider the sample core. Okay, if differential is too large, okay, cells will no longer line up in single file. Okay, maknanya sekarang ni kan macam ni. Okay, kalau dia punya ni, dia punya size ni, kalau itu dia punya size ni kan. Okay, kalau dia punya window tu, okay, if the window is too widen, okay, the window is too wide, okay, dia akan jadi macam ni. Okay, so what happened is that cells will no longer line up in single cell, okay, in single file. Maknanya cell tu dah tak go through in single file, okay, so dia akan go through in maknanya satu pass through tu dia boleh pass through sampai 5-6 cells, okay. So we don't want that to happen. Okay, if that ever happen, okay, then the data that you've got is going to be very inaccurate. Okay, so results in a wider CVs, okay, an increase in multiple cells passing through the laser at once. Okay, you will get faster data. Okay, you will get faster data, but you will lose the resolution. Okay, so what does it mean is you will lose the accuracy. Okay. Contohnya selalunya, okay, cell yang usually the amount of cell that we use to acquire tu maknanya the cell yang we prepare, okay, to be sucked into the Flowcyto, usually it is 100,000, okay, banyak, okay, especially kalau macam cell tu macam dari jenis yang PBMC, selalunya non-adherent cell dia kecil-kecil je. Okay, so non-adherent cell ni selalunya kita acquire more. Kita acquire up to 100,000 contohnya, or kita acquire up to 50,000 contohnya, okay. So when that happen, okay, usually it will take time. Okay, it will take time. But if you widen, okay, if you widen the size, okay, ni kan, you besarkan the window. Okay, kalau you besar, okay, so kalau you besarkan window dia, okay, akan ada banyak sel boleh lalu. Okay, but the problem is, okay, bila banyak sel lalu, of course lah, okay, your experiment will finish earlier. Okay, but you lost the resolution. Okay, maknanya you lost the accuracy of the data. So that is very important for you to understand. Okay, so this is what happened. Okay, so ini adalah low pressure. Okay, ini kalau dia adalah high pressure. So you tengok kalau low pressure nampak you dapat cantik je, nampak the peak is very beautiful. Okay, so meaning that this one shows that okay, only one cell pass through the file. Okay, kalau you widen it, you put high pressure, okay, can you see? Okay, dia punya peak dia jadi tak cantik. Okay, with increased sample pressure, flow rate increases, resulting in decrease in data resolution. So what does it mean by increase in data resolution? It means that the accuracy, okay, accuracy of the data, it can be, how should I say, it can be questioned. Okay, all right. Okay, so optic light scatter. All right. So when light from a laser interrogates a cell, that cell scatters light in all direction. Okay, so the scattered light can travel from the interrogation point down a path to detector. Okay, so this is what happened. Okay, so as I told you before, okay, when you have a laser beam, okay, and a cell, okay, that pass through, what happened is that the laser, okay, the laser that is the light, okay, emitted from the laser, okay, will reach the photochromes, okay, and then it will emit, okay, the scatter light. Okay, so what happened is that okay, dia akan scatter. So whatever that is being scattered, okay, will catch by the photo multiplier. Remember the photo multiplier? Okay, remember you ada laser, you ada cell, and then dekat sini dia ada photo multiplier. Okay, so this photo multiplier will catch, okay, all the scattered cells. Okay, why? Because if the photo multiplier fail to catch, okay, fail to pull, okay, all the lights, okay, nanti dia tak boleh nak convert it into scatter plot. Alright. Okay, so optic forward scatter. Okay, so this is what happened. You ingat tak tadi yang I cakap pasal shaft fluid? Okay, so this is where the shaft fluid will be. Okay, at this area. Okay, all right, so this one, okay, so these are the cells. So the cells should pass through, okay, in a single file. Only one cell, okay, only one cell at one time that can pass through the laser beam. Okay, so bila dia pass through the laser beam, okay, can you see the detector? Okay, so can you see the detector, okay, where it will catch, okay, all the light, okay, and then what happened next is at the electronic part where it will convert, okay, whatever light that has been catch, okay, into a scatter plot. Okay, senang kan? Okay, so interrogation forward scatter. So far ada masalah tak? Ada apa-apa tak faham tak? Ke dah tak boleh tahan dah, lapar sangat? Ke tengah makan? Ke tengah tidur? Saya dah habis makan. Tengah makan, okay. Alright, so interrogation forward scatter. Siapa yang tengah makan tu saya minta maaf saya tak berniat nak membantutkan you punya selera. Tapi kita punya lecture hampir habis ya. Tunggu delivery KFC, Alhamdulillah. Kejap lagi sampai, saya dah habis kelas dah. Okay, alright. Okay, so interrogation forward scatter. Alright, so light that is scattered, okay, in the forward direction, okay, along the same axis the laser is traveling, so it is detected in the forward scatter channel. Okay, so if you still remember, okay, dekat scatter plot yang tadi I show you, okay, the forward scatter is at the x-axis, okay, yang side scatter is at the y-axis, okay, so it is very important. Masa you plot the graph, okay, if let's say lah, ni kita ada soalan permasalahan, okay, or SAQ, you have to plot the graph. So it is very important to understand that this is the side scatter and this is the forward scatter. So side scatter will measure the granularity, okay, the forward scatter will measure the size of the cells, okay. So the intensity of this signal has been attributed to cell size, okay. So remember, bila you cakap pasal forward, dia mesti kena measure the size of the particles or the size of the cells, okay. Saya pun dah slurred dah ni, saya ada kelas berpagi lagi ni. Okay. Alright, so this is the laser beam, okay. So if you look here, this laser beam, they shoot at a single cell, okay, and then what happen is that dia akan bagi light, the light will be scattered, Okay, the light will be scattered. Okay, so this will be forward, and downward will be the side. Okay, so laser light that is scattered at 90 degrees to the axis of the laser path is detected inside scatter channel. Okay, so the intensity of this signal is proportional to the amount of cytosolic structure in the cell. So meaning that whenever you talk about side scatter, okay, it will only measure the granularity, okay, of the cell. All right. Okay, so it is very important. Okay. So why look at FSC versus SSC? Okay, dia kata kenapa you nak kena tengok FSC versus SSC. Okay. Since FSC, okay, is the size, okay, and SSC is the internal structure or the granularity, a correlated measurement between them can allow differentiation of cell type in heterogeneous cell population. Okay, so this is actually the reason, okay, why when you measure, you must look at both sides. Okay, you must look at the forward and you must look at the side scatter. Okay, why is that? Because certain cell, it may have similar size, okay, but different granularity. Okay, meanwhile the other cell, okay, might have similar granularity, okay, or cell intrusion, okay, but it may have different size. Okay, so that is very important for you to look at both sides, okay, because with both sides, okay, then only you can see, okay, then only you can really subpopulate, okay, the whole population of the cells. Okay, for example macam ni kan. Okay, so ni lymphocyte. So lymphocyte kita memang tahu, okay, selalunya lymphocyte tu memang ke arah bawah. Okay, tapi kalau if you look at the monocytes and the granulocytes, okay, there is one point where it can, how should I say, it can, alamak, apa lah yang perkataan yang paling sesuai, apa yang bertindan tu, what they call it, apa, apa, bertindan apa, it can overlap, okay, to one another. Okay, why is that? Because aspect boleh lah overlap. Okay, because kalau itu monocytes and granulocytes, okay, you have to also consider what happened to the live cells. Okay, live cells. Okay, selalunya live cells, debris, dead cells, okay, dia ada dekat area sini. Okay, dia dekat-dekat dengan lymphocyte. Okay, tapi dia bukan lymphocyte. Okay, so if you don't have, okay, the forward scatter or okay, the side scatter, okay, maknanya kalau tak ada dua-dua plot tu, okay, there's a possibility that you cannot really differentiate or do the subpopulations. Okay, only with the existence of these two, FSC and SSC, okay, then only you can really differentiate the subpopulation. Okay, kalau you tengok betul-betul kan, ada satu, ni slide berapa kita kita tengok yang tadi yang bergambar tu. Mana slide gambar tu? Okay, so if you look at this slide, okay, you can see there is some overlapping. Okay, perasan kan? Okay, dia ada overlapping. Macam monocyte ni kan, dia boleh overlap, okay, dengan neutrophil. Okay, kalau macam eosinophil pun dia boleh overlap. Okay, dia boleh overlap dengan basophils. Okay, for example. Okay, but as I said, the most important thing is you have both, okay, the FSC and SSC. If you have only one, okay, then it is very hard for you to differentiate the subpopulation. Okay. All right. Okay, so let's look at the optics, okay, or what we call detectors. Okay, so there are two main types, okay, of photodetectors, okay, used in a Flowcyto, okay, where you have the photodiodes, okay, and photomultiplier tubes, okay. So the photodiodes, okay, used for strong signals, okay, when saturation is a potential problem. Okay, photomultiplier tubes or PMT. Okay, this one is more sensitive than photodiode. Okay, so the PMT is used for detecting small amount of fluorescence, okay, emitted from fluorochrome. So this usually the PMT ni dia adalah untuk it is for the use of a very sensitive experiment. Okay, very tedious experiment selalu kita pakai PMT. Alright. Okay, optic filter. Okay, so oh, Lisa dah left ke? Dia pergi cuci tangan ni, dah habis makan. Tak apalah, dah nak habis dah pun. Okay, so optic filter, so different wavelength, okay, of light. Different wavelength of light, okay, are scattered simultaneously, okay, from a cell. So what happened is it need to split, okay, the light into its specific wavelength, okay, in order to measure and quantify them independently. Okay, so this is done with filter. So optical filter, okay, they are designed such that they can absorb, okay, or reflect some wavelength of light, okay, while transmitting the others. Okay, so this is actually the important part of the optic, okay, or filters, okay, where usually, okay, there are three types of filters, okay, where we have long pass, short pass, band pass, and dichroic. Okay, so I think I'm going to stop until this slide, okay, where you have more of the filters, okay, ahead. Okay, so I'm just going to stop until here, okay, and we're going to continue. When? You want to continue tomorrow after the test? Nak sambung esok tak lepas test? Ada presentation. Okay. So presentation you pagi ke bu? So when is your presentation petang. Okay, okay, okay. Okay, so I think we're just gonna continue next week lah. Okay, kita continue next week saja, boleh? Okay, alright. So for tomorrow, the test will start at 10:30. Boleh? 10:30? Esok test start pukul 10:30. Boleh? You ada kelas ke sebelum tu? Boleh. Alright, okay. So tomorrow, okay. Alright, so tomorrow the test will start at 10:30. Okay, dia punya duration 1.5 hour. Senang sangat dia punya ujian. Okay, cuma ada 5 saja soalan. Okay, you'll definitely finish it on time. Okay, so I rasa itu sahaja. So we'll stop until here. Okay, and we'll continue next week. Okay, so I think that's it. Doktor nak tips? Tak ada tips. Okay, tips tu sangat senang. You baca je, mesti you boleh jawab. Okay? Okay, alright. Thank you to you too. Doktor nak, alamak, recorded. Ya Allah, sorry, saya tak record lecture hari ni. Sorry, saya tak record lecture hari ni, sorry. Tak apa, okay, thank you. Okay, bye-bye. Selamat
Okay,
makan.
bye doktor.
Mana dapat KFC tu, makanlah dengan seronok ya. Okay, bye. Assalamualaikum.
Waalaikumsalam.
Thank you, doktor.
Okay, bye. Eliza punya apa tu? Eliza punya apa tu?
Recording kot.
Recording?
Mm-mm.
Okay, nanti saya cari balik. Ada. Nanti saya cari balik ya. Tapi soalan dia sangat senang. You pejam mata pun you boleh jawab. Okay,
Macam
bye.
takut je.
Apa dia?
Macam takut je nak jawab test
Tak
esok
lah.
ni.
Tak, senang je. Saya baik lah, jangan risau. Senang je. Esok pejam mata typo macam. Okay, dah. Ain,
Okay, okay.
pergilah makan KFC. Siapa order KFC tadi? Okay, bye. Saya live ya. Bye, Assalamualaikum.
Hai Gilang. Dah live